So moving forward with the blood, we'll look next at leukocytes and platelets. This term leuk refers to white. So these are white blood cells. And platelets we'll see are also sometimes called thrombocytes. So revisiting this slide, we're reminding ourselves that if you take whole blood and spin it down, you will end up with something called a buffy coat. Not the super light, watery plasma, but not that heavy iron-laden erythrocytes. In the middle, you've got these moderately heavy and dense leukocytes and platelets. So leukocytes first. The function of these leukocytes is they play a role in immunity. So defending against disease, and that's done in a lot of different ways that we'll talk about. They are the only formed elements that are complete cells because platelets are just little fragments of these bigger cells, and erythrocytes are missing many organelles and they eject their nuclei so that they can carry more hemoglobin. So leukocytes are the only intact cells. There's not many of them, less than 1% of total blood volume is leukocytes, and they do this interesting thing called diapedesis because unlike red blood cells, these white blood cells need to go and fight infection anywhere it may be found in the body. So that means they need to exit the blood. And what will happen is in the case of some type of infection, some of the early immune cells that arrive will release these signals. And I'm not getting into a lot of detailed terminology here, it's not really part of the class, but these signals will eventually make their way into a blood vessel and create what I think of as kind of a like runway lights. As these white blood cells are speeding past, if they see these little signals, these little lights, they will know that they should land there, crawl or ped is like foot to walk, like a pedestrian. So they will walk themselves down the edge of the blood vessel and squeeze out in between some of the epithelial cells, and then they can make their way to the site of the infection. That process is called diapedesis. There are 5 types of leukocytes: basophils, eosinophils, lymphocytes, monocytes, and neutrophils. Here, these 5 are in alphabetical order, and you could come up with some mnemonic device for that, but there's actually a better way to sort these using the mnemonic "never let monkeys eat bananas." That provides the relative abundance of leukocytes. It just means which one of these is the most common and which are the least common. So neutrophils are the most common of all your white blood cells, down to basophils, which are the least common. Never let monkeys eat bananas. And so as we go through the leukocytes over the next few slides, I'm going to tackle them in this order. But there's one other way you could have sorted these, which we'll talk about, and that's if you notice the terms here, 3 of these are ending in "phil," and sometimes they're referred to as the 3 phils, where the other 2 end in "site." So the 3 phils end in the term "phil" because "phil" means to love. So like a bibliophile loves books, and Philadelphia is the city of brotherly love. These 3 phils love stain. So when we look at histological preparations, typically you see these pinks and purples because there are these 2 different stains that are applied to tissues to stain either more acidic or more basic structures. So these 3 types of cells, these 3 types of leukocytes, they absorb the stain. They love the stain. There's a specific part of the cells that love the stain called granules. So the 3 phils are also called granulocytes. Granulocytes. That means they have these granules. The other 2, lymphocytes and monocytes, lack granules. So they are agranulocytes. So then the question is, well, what are these granules that are absorbing these different color stains? And so you can get very detailed with this and talk about the myeloperoxidase, the serine protease extracellular trap. We're not going to get into all that. This is just to show us that this is just describing the granules in neutrophils. And then there are other types of granules in these other leukocytes. You can learn about this type of stuff in an immunology course. For our purposes, all we care about is the fact that each of these leukocytes fight infection in different ways. Some of them have special granules to help with the fighting, and they absorb these stains in certain ways. So we don't care about the details, we just know that some of these cells have granules that are involved in making the weapons that they use to fight infection. So let's look at them each in order, in order of their relative abundance. Never let monkeys eat bananas. N is for neutrophils. Neutrophils, let's look at the name first. Neutrophils are kind of neutral in their love of the stains that we apply to these tissues. So like if you look at this picture, you see there's some purple and there's some red. So those are the two stains, are these H and E stains. One is purplish and one is reddish. Neutrophils, they have granules. See those little dots inside the cell are granules. That's cellular machinery. Some are more purplish and some are more pinkish. That's why they came up with neutrophil as the name, because they're kind of neutral. Some granules are absorbing the pink, some absorb the purple. These are the vast majority of your leukocytes. They're fairly large compared to a red blood cell, but the major way to identify these is through the nucleus. This is their nucleus, multi-lobed. In this case, I see 4 separate lobes. They are attached to each other, so you may see some strands connecting the lobes. And usually you can like imagine some sort of funny face inside the cell from the nucleus. Yeah, I can see it a couple ways. So each of these types of cells, we're going to ask ourselves, how are we going to tell it apart from the other leukocytes? And here, you can use size a bit, but really that multi-lobe nucleus and the neutral color, it's not super purple out here, it's not super pink, it's kind of both in the middle somewhere. Those are the ways you're going to identify neutrophils. What's the function of neutrophils? Phagocytosis. They are going to go out and there's tons of them, right? And they're just kind of roaming through your body and they just eat up just about everything that they find. Lymphocytes are next. So pretty common, maybe a quarter of your leukocytes are lymphocytes. They have this dark circular nucleus. So very different than that multi-lobed nucleus from neutrophils. In some ways, you'll hear this described as that you'll know it's a lymphocyte if you see a halo of cytoplasm or a ring of cytoplasm. Now keep in mind, this nucleus is a little bit pushed to the side, so it's not a complete halo or a complete ring, but more or less you can see it there. So these lymphocytes are mostly found in your lymphatic tissue, which we'll cover soon in this course. Some of them are cruising around in the blood, but a lot of them are in these big clusters in certain places in your body. And the function of lymphocytes is to provide what we call specific immunity. And we'll be talking about that more later when we discuss the immune system. But what we mean here is that a neutrophil is just kind of this like brawler. It's a big, beefy fighter that goes around and whatever it encounters, it's going to attack. Lymphocytes are more specific. So if you get a flu shot, what you're doing is you're training your body to fight against these specific strains of flu in that shot. Right? It doesn't have all flu strains. It's just the ones that the CDC predicts will be the most common this flu season. So they're going to only fight specific strains of virus or bacteria, etc. Some of them also produce antibodies. We'll talk about that more later. Monkeys. Never let monkeys eat bananas. So M is for monocytes. Getting more rare here. Less than 10% of the population are monocytes. I think of them as monster cells because they're really large. And also that U-shaped nucleus or kidney bean shaped nucleus reminds me of like maybe some little alien or Godzilla fetus growing inside an egg. It's a monster site, a monocyte. What is the function of these monocytes? They will also be discussed with the immune system in more detail because they will leave the circulation, go into tissues, and then they change and they become these macrophages, which means big eaters. So they're actually going to work with the lymphocytes to fight against some specific strains of invaders in your body. We'll talk about that more later. Eosinophils. Eos, just like erythro, refers to dawn, like a sunrise. So in this case, we're talking about the red coloration, like a red sunrise. So obviously that's something you want to look for right away. You can really see those individual granules that have absorbed that red stain. But, sometimes the stain wasn't absorbed as well, or the slide is old, or whatever. So that red is not always the best indicator, but this bilobe nucleus is also good. Sometimes you'll refer to it as a pair of lungs, right? Because they're roughly shaped like lungs. There should always be two. And if you're looking at something and you think, I think it's red, I think it's an eosinophil, check for the lungs, and that should confirm. The function of eosinophils is to counterattack against parasitic worms. And if you haven't gone on YouTube and looked up the videos of people having these worms removed from their eyes, that is worth a watch. Last is basophils, very rare. They do have an S-shaped nucleus, but typically you're not going to be able to see it because these granules are stained so dark. Base refers to the more basic stain. This purple stain stains things that are more basic. Like nuclei get that dark purple stain, but it's so too in basophils do the granules get that dark purple stain. So I can't really make out the nucleus. The job of basophils is releasing a couple chemicals that are involved in the immune system. Histamine dilates blood vessels. Heparin limits clotting. So histamine, you may not know much about histamine, but you probably know about antihistamines. Like if you have some sinus pressure, in your face, and you take an antihistamine, you're trying to relieve that pressure. And the pressure is there due to inflammation. These blood vessels dilate because you've got some sinus infection, and there are immune cells fighting and dying in there and attacking substances, creating all this like waste products. That inflammation is going to bring extra blood there, and it's going to help clear out this battlefield. And more blood means more white blood cells are arriving at the scene to help with the fight. So that's the purpose of inflammation. If you take antihistamines, you are kind of taking away one of your immune system's weapons. But, if it also takes away your discomfort, then who cares? Yeah, that's what I would do. Heparin prevents clotting, meaning, well, we do want to keep that blood moving in the case of an infection to arrive at that battlefield and to bring the white blood cells and to carry away the waste products. So heparin can assist with that. So, where do all these white blood cells come from? Well, if you remember learning about where red blood cells come from, the answer was that there are pluripotential stem cells found in red bone marrow, and all those blood cells are made there. So leukocytes are also made in the red bone marrow from pluripotential stem cells. We learned this term hematopoiesis at the time too, meaning the creation of blood cells. And then we learned that you can say erythropoiesis is the creation of erythrocytes. And so to leukopoiesis is the creation of leukocytes. And if you want to get really detailed, you could say lymphopoiesis is the creation of lymphocytes. And in fact, that has been studied a lot. Like there's just one type of lymphocyte called B cells. And look at all the different pathways and intermediate stages that you can take just before you get a type of B cell. B cells make antibodies. So it's very complicated stuff. And again, this is a immunology type of stuff here. We're not really concerned with all that detail. But this is why we're learning these more specific terms, because they really do open windows into very complex processes. And here's some references again to the lymphatic system, which we'll be talking about soon. And we'll finish with platelets, which are also called thrombocytes. Platelets are small purple staining cell fragments, and they come from these gigantic cells called megakaryocytes. So here's a little red blood cell. Here's this massive megakaryocyte. All these tiny little platelets are flaking off. Platelets are involved in clotting your blood, which we call coagulation. And they're one of two structures that you should know about in regards to clotting. The other being those fibrin strands that come from the fibrinogen proteins floating around in your plasma. They're going to work with the platelets to form, oh, there's a megakaryocyte, supermassive. But the platelets will work with the fibrin to create like this net or this web, and you'll be trapping red blood cells and trapping white blood cells and stuff in this web, and this is how a blood clot is formed. Blood clots are also called a thrombus, which is why it's kind of nice to know that the other name for platelets is thrombocytes. Because then you'll know anytime you hear a reference to thrombus in medicine, that's referring to thrombocytes, to platelets. So this has something to do with clotting. And in fact, a thrombus is just the name for a blood clot. And why do we care about blood clots? Well, because when blood clots go bad, you can have a lot of pretty bad consequences. So like a stroke is what happens when your brain or portion of your brain stops receiving blood because you had this clot form, and then the clot broke off and whooshed its way through the blood vessels and eventually got into your brain and got lodged in a blood vessel in your brain. And that's when we call it an embolus, is when a blood clot travels through the vessels and then gets stuck and becomes stationary. It's an embolus. So obviously that's pretty bad news. Another term you may have heard of is deep vein thrombosis, DVT. This is a pathology that you want to prevent on like long plane flights by getting up and walking around, and there's little exercises you can do and stuff. You would think when you learn about these things, you would do them, but I don't do them. But you should do them. You should exercise on that plane, because what can happen is when your legs are all compressed in these weird positions and not moving much, you can start to form these little clots on the inside of your veins. And the veins bring blood back to the heart and then push it into the lungs. So any blood going through your veins is going to be on its way soon to the lungs. So in this case, you could form a little clot, a little thrombus. That could end up in your lungs, block a blood vessel, and now you've got like a pulmonary embolism. Now you've got a big problem that we won't get into here. So those are leukocytes and platelets. They make up the buffy coat of spun out blood. And we've now covered all of the components of blood. We'll finish with a look into blood types. If you've covered this in microbiology, that's great. You'll see more about it in that class or in physiology. We're just going to do kind of an introductory look at blood types, including antigens and antibodies.